Well, so let me first then thank the organizers for the opportunity to talk to you here, and Michal for that very kind introduction that almost pointed out all the highlights I wanted to bring here. Let me also start out then by pointing out the very obvious to you, and that is that if you're interested in human origins and in human history, the human genome is a great source of information, and also the fact that what we do, of course, normally when we want to reconstruct the history of the human genome is that we compare DNA sequences of people that live among us today. And as you know, we find a difference every 1,000 base pairs or so if we compare people in this room. If we go and add a chimp, we find a difference maybe every 100 base pairs. And we test models we have for how DNA sequences change over time, reconstruct the history in the past that have given rise to the differences we have today. And we generally depict that then in the form of a tree like this. But of course, this is a quite indirect way to study human history or the history of the genome, because we reconstruct by what we see today, what we think has happened in the past. So, let's see if this guy. So what I then want to describe to you is sort of our attempts since over 20 years now to, in some cases at least, go back in time and try to retrieve genomes from extinct ancestors or close extinct relatives of people today. And if you just are quite naive about it and look on things such as ancient Egyptian mummies that look very well preserved, you might think that this should be rather easy. But if you then try to look in tissues from such remains, for example, by microscopy, and compare here a contemporary human muscle to a 3,000-year-old ancient Egyptian muscle, you will see it's a very depressing picture, actually. Here, the muscle fibers, cell nuclei where the DNA would be, Egyptian muscle, there are no intracellular structures, no cell nuclei. But in some rare cases, there are anecdotes. This is the first mummy where this worked, a 2,700-year-old mummy from Egypt, from the skin here, where you can stain cell nuclei with dyes specific for DNA in the skin. If you then go and extract such DNA, what you will find is that it's degraded to short little pieces, 50, 60, 70 base pairs generally, and it has chemical modifications that may cause errors in the sequences you determine. So if you want to reconstruct a longer piece of DNA, what you have to do is cumbersomely retrieve short overlapping pieces, repeat your experiments many times to make sure you're not fall victim to these chemical modifications here. So because of all this technical, most of the work that has been done to date have been done on mitochondrial DNA, simply because of the fact that there are hundreds of thousands of mitochondrial genomes per cell, so that there is much bigger chance that at least some of them will survive. And it's pretty routine today then to do this from zoological collections that have been collected over the last few hundred years for animals that are recently extinct and extinct during and before the last glaciation. But our particular interest are then in Neandertals. And as you know, Neanderthals are this robust forms of humans. Here's a Neanderthal skeleton compared with the contemporary human skeleton that appear in the fossil record somewhere, depending on how you define a Neanderthal, between 2 or 3 or maybe 400,000 years ago. They live in Europe and in Western Asia until they become extinct around 30,000 years ago, and then become replaced by fully modern humans, humans with a skeleton identical or almost identical to ours. And there are two general ideas around today about the relationship of present-day humans to Neanderthals in Europe and other archaic forms in Asia here. One is that modern humans come out of Africa, starting somewhere around 100,000 years ago, and replace Neanderthals without any admixture at all here. The other one is that modern humans do come out of Africa, replace Neanderthals and these other forms in Asia, but there is gene flow from them into present-day humans. And these two ideas then, of course, give very different predictions about what a Neanderthal DNA sequence or genome would look like. In this case, the Neanderthal would be equally distant from everybody today, no matter we live in Europe or we live in Asia. In this case, it would on average be more similarity to at least some people in Europe. So the first chance to test this then came obviously by studying nuclear DNA, and not just from any, from mitochondrial DNA, and not just from any Neanderthal specimen, but we were very lucky. The first samples we got came from this specimen, which is a type specimen, which was found in 1856 in Neanderthal and gave their name to this group of humans then. So I'm relaxed here, since there are no paleontologists in the audience probably, so I can probably say that I think we were particularly lucky that we got a type specimen to start with, because paleontology is a very difficult discipline, I think. So whatever Neanderthal specimen we have since studied, there had always been at least one paleontologist who came to us and said, it's not quite a typical Neanderthal, it's a little too gracile, a little robust or something, but if this is not a Neanderthal, then Neanderthals don't exist, so to say. So I came from the upper arm here, studied a particularly variable part of the mitochondrial genome. As we said, cumbersomely retrieving overlapping pieces, cloning them, looking at many clones, seeing what is conserved among all or almost all clones, and estimated then a phylogenetic tree for the mitochondrial genome of Neanderthals. And they go back to common ancestor, mitochondrial ancestor, then a bit over 500,000 years ago, whereas all human mitochondrial DNAs today go back 100, 200,000 years. Since then, a number of other mitochondrial genomes have been determined. They all fall together outside the variation of present-day humans. So it's very clear Neanderthals did not have mitochondrial DNA to people today. But that's then the mitochondrial situation. We know, of course, that allelic differences among people that live today go back, on average, something like 500,000 years, and often more, up to 1 million years or even more. So if we now from the mitochondrial data know there was a split somewhere after 500,000 years ago between Neanderthals and modern humans, we know already without studying anything in the nuclear genome that some people today, for some part of their genome, must be closer to Neanderthals than other people today, just because the variation today is deeper than the population split to the Neanderthals. So that then means that the Neanderthals cannot have difference from us. This is an attempt to illustrate that. And it would be particularly interesting, I think, to study their nuclear genome, because this situation is then typical of average parts of the genome. If something special happened in humans after we separated from Neanderthals here, some mutation happened, was positively selected, and swept through to everyone today, there the situation will look like this. We will all go back to common ancestor, falling outside the variation of Neanderthals. So by then studying this, we should be able to sort of focus on the most interesting substitutions, perhaps, that happened recently in human history since we separated from Neanderthals. And they might be particularly interesting because among these, most of them will, of course, have no functional consequences. But among them will presumably hide some things that made fully modern humans possible. And it's only with fully modern humans that some crucial things happen in our history. Technology comes, technology that changes rapidly, becomes regionalized in the world, and takes off almost exponentially. Art appears. And today, immediately, intuitively recognize it. But then that needs to be the nuclear genome of the Neanderthals. And clearly, until 5 years or so ago, and I think almost everyone else thought that this would always remain impossible. But of course, as so often, it's really technology that makes such statements that things will remain impossible, obsolete. And in this case, it's high-throughput DNA sequencing that made that. The fact that we can now sequence millions of DNA molecules very, very rapidly and at lower and lower costs. So what you can now do is not try to retrieve particular parts of the Neanderthal genome by PCR or so, but simply make a DNA extract from the bone, sequence all the molecules that have there as random molecules, create your little database, and start comparing it to the human genome, the chimp genome, bacterial genomes, and so on. And the first place where this worked was this cave in Croatia, India, and from this bone here, and actually this little part of this bone here, 38,000 years old. And if you look at the DNA fragments that you retrieve here, they're indeed very, very short. Average size, something like 55 base pairs or so, hardly anything above 150, 160 nucleotides. Something else you will notice is that only a tiny fraction of the DNA actually comes from the Neanderthal. So in our very best extracts here, around 3% or a little more come from the Neanderthal. The entire rest are from microorganisms, bacteria, fungi that colonized the bone after the death of the Neanderthal. So we spent the last few years looking through a lot of sites where you find Neanderthals, a lot of bones, to find the best ones, particularly in this ratio of endogenous Neanderthal DNA relative material DNA in them. We identified 3 bones here from Vindija from 3 different individuals that have now contributed almost all the data we had to analyze. In addition, we have identified 3 other sites that also contain usable DNA. One is a type specimen site, one a site in Spain. They're all around 40,000 years old, and an older site here in southern Russia, 60 to 70,000 years. So we worked a lot technically in the lab in the last few years on making the process with which we come from something we extract from the bone to sequencing library that we can put into the sequencing machines more efficient. So we're about 500-fold more efficient than a few years ago. The machines became more efficient in terms of numbers of molecules they can sequence. They also allow longer molecules to be sequenced, but that's not so interesting for us then. So in the end now, the 3 billion base pairs we just published come from a total of around half a gram of bone. So it's not enormous amounts of bones you have to use for this, but you have, of course, screened a lot of bones to find the best ones to do this. So we sequenced a bit over 3 billion fragments, most of them then from bacteria, but retrieved something like 3 billion base pairs, approximately equally distributed among from these 3 individuals from Vindija, very little from the other sites so far. So the average genome coverage is something like 1.5-fold. So there are a few technical things just to point out. The big risk when you do this is, of course, contamination with present-day human DNA from yourselves or from chemicals, from paleontologists that have handled the bones. So you work under clean room conditions, and we have different estimates of the level of contamination in the actual data put out. So one is based on the mitochondrial genome, where Neanderthals do differ from all people living today. So we can distinguish mitochondrial DNA from Neanderthals from people today. The estimate of that is something like 0.3%. We are lucky in that these 3 individuals we study are females. So we can look at Y chromosomal DNA as an estimate of male human contamination to the data. And the estimate of that is something like 0.5% or a bit more. And we also use an approach where we identify in one bone positions that look ancestral, where as far as we know all humans today are derived, and use a second bone then to estimate how often that is derived and ancestral. And it's, of course, confounded by polymorphism among Neanderthals and contamination. So there's a likelihood approach to estimate that, which is also below 1%. So when you look at any particular position in the Neanderthal genome, there's a bit below 1% risk that is actually contamination from present-day humans. When you then align the Neanderthal fragments to the human genome, you will see differences. And you will see quite a suspicious pattern if you just do this. You will see particularly many differences in the beginning of fragments and in the end of fragments. And in the 5' end of fragments, these will particularly be C to T differences where the Neanderthal has T. In the 3' end, G to A differences where the Neanderthal has A. So you can see this here too. How C to T start very high and go down rather rapidly, and G to A in the other end. So this obviously looks like an artifact. And we know what it is. It's deamination of cytosine residues that then lose this amino group, become uracil, which are then read by DNA polymerases. And this happens particularly in ends, we think, because where the DNA is single-stranded, it's particularly susceptible to deamination. And we then make blunt ends with an enzyme, T4 polymerase, which leaves the 5' ends intact. So there we really see the C to T changes. But overhanging 3' ends are shooed away, recessed ones are filled in. So in the 3' end, we see the reciprocal G to A changes in what we then sequence. So quite a lot of bioinformatic work, particularly done by Ed Green in the group, have gone into then approaches to align these short sequences with these particular error patterns to the genomes of, say, humans and the chimpanzee genome. We often align to the chimpanzee genome to avoid biases depending on what human genome we align to. So the average coverage, as we said, is just a bit over 1-fold. So if you look in the brown now, in any particular position you're interested in Neanderthals, you have something over 55% chance to actually see that position by at least one read. So you can begin asking overall questions. One thing may be, how divergent are Neanderthal DNA sequences from present-day humans? So since we have low coverage and lots of errors in the Neanderthal sequence, we don't just look at differences between humans and Neanderthals. We say at positions that changed on this lineage, where humans differ from chimps and other apes, how many of them do the Neanderthal look like the chimp? So it happened here. How many of them do the Neanderthal look like humans? So the change happened here. So we ask essentially how many % back along this lineage with chimp do sequences diverge? The answer is 12.7%. If we say this is 6.5 million years, this is something like 800,000 years back to the average common ancestor of DNA sequences. We sequenced also 5 modern humans in this study to get a perspective on this. A typical European, which for us, of course, is a French person, that's 8% back, around 500,000 years. Chinese, Papuans, very similar. And among the deepest divergences we think we know of in Africa, approach 700,000 years. So the Neanderthal is on average, say, 20% or so deeper than the deepest divergences we know about in humans today. So quite close still. What you can now begin to do also is assemble a catalog then of all features in the human genome where we differ from the Neanderthal. The Neanderthal looks like the chimps, and as far as we know, everyone is fixed today. So this is an interesting group of differences where something spread rapidly to everyone who's around today. So we look for fixations here, which are then quite rare. They are indeed quite rare. If we look at inferred amino acid substitutions, we find only 78 such amino acids. Now, this is a number that's total in flux. This is what is in the paper. Of course, we have only about half the genome, so this will double because of that. But through the 1,000 Genomes projects and other sources, we of course learn low-frequency variants quite rapidly in humans. And quite a few of this turn out to then exist in low frequency still in humans. So I think it's somewhere down a little below 50 now in just a few months since the paper appeared. But so you can look at these lists. It's hard to make big sense out of them. But for example, if we look at those two fixed amino acid substitutions or start-stop codon differences, since we separated from Neanderthals, there are only 6 such proteins. And maybe it's interesting that 3 of them are actually expressed in the skin. So maybe that's that skin physiology or morphology changed rather really in human history. What's particularly interesting is, of course, to find evidence for positive selection. And positive selection then in humans since we separated from Neanderthals. And to do that, we can take advantage of this fact that I pointed out in the beginning, that the Neanderthals fall into our variation for most of the genome. So if we look at SNPs in humans today, we know the ancestral state, of course, from the apes, and we have a derived state, in this example, a T. And quite often will the Neanderthal have the derived variant, meaning falling into our variation. If we just ascertain SNPs from these 5 genomes we used, this happens in 10% of SNPs in humans today. But if a selective sweep happened afterwards, since we separated from Neanderthals here, there, of course, per definition, the variation is more recent, so the Neanderthal will always fall outside, should in principle never have the derived variant, except for parallel mutations. So what we'll do is look for SNPs. Here in humans, we have the chimp genome, we have a Neanderthal genome, we'll have a window. In 10% of cases in any one window, will the Neanderthal have the derived variant? We would look for areas like this, where suddenly the Neanderthal look all ancestral, because there something may have happened. So you can then have a measure for this depletion of derived variants and for how wide these regions are, genetic width. Because of course the stronger the cells, the faster it swept through the population, the less time was there for recombination to make this region smaller. So the most strong sweeps with the strongest selection will be the widest ones. So you can then look at the things that are up here that have very few derived variants or hardly any and are particularly wide. So the most extreme case in this first version of the screen is a gene called FADA. And you can start, and this is a list of the top 20 candidates there. Some of these regions then have no genes in them, suggesting that it might be something regulatory or so that have changed there. We can get some idea about what genes this may be involved in by looking at disease mutations that are known for them. So for FADA, for example, it's a risk allele for type 2 diabetes. It's quite interesting. It was perhaps not something well expected. Maybe it hints that something in metabolism or so have changed in humans since we separated from Neanderthals. We can also get a feeling for about where the causative changes might be to be looked for. So if we look, these green dots are derived SNP alleles in the Neanderthal. We see this area with a paucity of derived variants, and it's particularly extreme here in this intron. We can focus in on it, and in that area then there is a conserved 170 base pair sequence conserved among all mammals, where humans have an insertion of 9 base pairs that's lacking in the Neanderthal. So when we published the paper, we thought this was fixed in humans. In the meantime, we know that 3-4% of people in Europe actually have non-inserted variants, so the Neanderthal-like variant. Interestingly, those are the ones that are protected against type 2 diabetes. It is a risk variant for diabetes. Fascinating to speculate about this, that something might be positive here. We don't really know what is positively selected, but maybe the course we pay is our tendency for type 2 diabetes when we have access to food. We can look at other things on this gene. Maybe it's of interest that 3 of them are involved in cognitive development in different forms, autism, schizophrenia, and the mental retardation in Down syndrome. I can't stop myself from showing you one gene that is particularly sort of tempting to speculate about, and that is the gene RUNX2. Mutations in RUNX2 are responsible for all cases we know of a rather rare syndrome called cleidocranial dysplasia. So it's a problem with skeletal development. The symptoms are here. I just want to point out 3 of them something called frontal bossing, problems with the development of the clavicle, and a bell-shaped rib cage. So first frontal bossing is these protruding frontal bones here. And of course, a more protruding frontal bone is a difference you see between modern humans and Neanderthals. The result is this sort of famous brow ridge here. The clavicle is one of the few bones that has a dramatic difference in form between Neanderthals and modern humans. Neanderthals have ancestral variants. Modern humans have something derived. It has been suggested by some that Neanderthals could not throw overhand because of this difference in shoulder architecture. They would throw like girls, like this, and like chimpanzees. And a bell shape here in a CCD patient compared with a normal control is a difference between the Neanderthal skeleton and modern humans. So it's very tempting to speculate that some change in RUNEX2 have to do with things that we actually observe in the fossil record. So all this is speculation, but now at least we have these genes and we can begin to start looking for the causative variants here. Another thing that is of interest is then what is actually the relationship between Neanderthals and present-day humans here. So what happened when modern humans start coming out of Africa, starting maybe 100,000 years ago or so, when they met Neanderthals? Did one interbreed or not? So our bias from the mitochondrial work was certainly to think there was no interbreeding. But so we sort of took three approaches to say, if there was interbreeding, what would we expect? So first expectation then would be that we would find some deep genetic divergences in Eurasia that would come from Neanderthals that we would then see in our Neanderthal genome. So Rasmus Nielsen at Berkeley particularly did this. He looked for regions in present-day humans without looking at the Neanderthal at all that look like this, where you have a clade of sequences that you find in Africa and outside Africa, and in addition, a deeply diverged variance that you see only outside Africa in the data we have available at the moment. Then identify SNPs that sort of tag this variance and look in the Neanderthal. What do the Neanderthal have at these positions? And among the 12 top candidates for things that are a lot deeper outside Africa than inside Africa, an unusual pattern in the human genome, right? 10 out of 12 of them are seen in the Neanderthal genome. Since most of them are actually at low frequency, 5, 6, 7% in humans, this is a very unlikely result unless these regions sort of come from Neanderthals into humans. The second expectation was that if there was interbreeding with Neanderthals, we would expect to see outside Africa, in Eurasians, Neandertal haplotypes, things that would be particularly close to Neanderthals in some humans, but not all humans outside Africa. So we look at that first to the African parts of the human reference genome, where we expect no contribution from Neanderthals since they were never in Africa. So if we plot here then how different one is from the reference human genome and plot how far one is from another human, Craig Venter's genome, that we believe to be a fully modern human. So if we have a lower and lower divergence to the reference genome, you tend to have lower and lower divergence for the regions in the genome also to Craig's genome. As if this is just a question of lower and lower mutation rates, because as you're closer to the Neanderthal, you're also closer to other humans around today. If we do this now for the European part of the reference genome, we see a very different picture. As you get closer and closer to the reference genome, you get closer and closer to Craig, except when you get very close to the Neanderthal genome here, you suddenly get very different from Craig, as if this is something that has come into some people, not because of low mutation rate now, but because it came from some other source. So the third line of evidence is in a way the most direct one, saying that if there was gene flow from Neanderthals, we would expect some Eurasians on average to be closer to Neanderthals than to Africans. So this is this approach again, saying if there's no gene flow, Neanderthals should be equally different from Africans as people in Europe. If there is gene flow, on average closer to. So we just look among our 5 individuals we sequence, for example, at positions where a French individual differs from an African individual. We look at derived alleles in Neanderthals and say, do they have the French or the Yoruban variant across thousands and thousands of such positions. So if we do this by comparing 2 Africans to each other and look at the difference in allele sharing between the Neanderthals and 2 Africans, it's not distinguishable from 0. If we do this between 2 non-African genomes, it's indistinguishable 0. But if we identify these positions between an African and a non-African, we see consistent more similarity, 4 or 5% more similarity to the non-Africans. And the disturbing thing here, or surprising thing here, was that we see this not only to the French, but to the Chinese individual, to the Papuan individual, so areas of the world where there have never been Neanderthals from the fossil record. So the surprising thing is then that we see this also in China, we see this also in Papua New Guinea, for example. So the idea we suggest for this is that when modern humans come out of Africa, maybe 90,000 years ago or so, the first place where they meet Neanderthals is in the Middle East. And if the interbreeding then happened there in modern humans that then became the ancestors of people everywhere outside Africa, that genetic contribution would sort of have traveled with these modern humans to these different parts of the world where there had never been Neanderthals. So that today then about 1 to 4% of the genomes of people outside Africa derive from Neanderthals. So this has been very fascinating for people that the Neanderthals live on a little bit. And there has been quite a lot of people since the paper came out that has been writing to us, self-identifying as Neanderthals. And I had to share with you some statistics that I compiled just the last few days among those people who write. It's quite surprising, actually, that of the people that write, almost everybody who identifies a Neanderthal is a man. It's very rare with Neanderthal women. And it actually seems that it's only Neanderthal men today that contribute genes to the next generation too, because if we look among the people who us and say that their spouse is Neanderthal, it's almost exclusively women that do that and no men that do so. So there is some interesting genetic patterns clearly going on here that I will then not go further into. But so rather for the last few minutes, what I wanted to do is to discuss where do we go from here, what sort of we're going to do in the future. The first thing that will happen is, of course, quite obvious, and that is that we need a much better, or want a much better Neanderthal genome. So we need to come to something like 10, 20-fold coverage over the next year or two. We're working on technology to that, to sort of fish out the Neanderthal DNA from these complex mixtures in various sources. So that's something rather obvious, technical, that will happen. The second one is a general problem with this field of research that I want to bring up here. And that is that we identify all these interesting candidate genes, such as RUNX2, etc. But how do we take the next step and actually prove our point and show that some change, specific change here, is actually of functional importance? So we need sort of tests, functional tests, and I want to illustrate that then with one gene we're interested in over the last 6-7 years, which is this gene FOXP2 on chromosome 7, which was identified by Tony Monaco's group in Oxford in a pedigree, 3-generation pedigree of a severe language and speech problem. And as you see, this segregates as if it was an autosomal dominant gene, and it is, and they found it, and it encodes this gene, transcription factor gene FOXP2. It encodes a very conserved protein, but it has 2 amino acid changes between humans and apes that happen in one part of the protein here that one doesn't really know what it does. So we were, of course, very interested in looking in the Neanderthal what they had at these 2 positions that change in the FOXP2. So comparing humans and chimps, these are the 2 positions that differ. This is the Neandertal reads that cover that in the genome sequence. And I must say, to my surprise, they turned out to be exact present-day humans. So the Neanderthals were similar to us in having these two amino acid substitutions. So they happened further back here before the separation from the Neanderthals. That still makes them functionally interesting, of course. It's interesting to ask, do they have any consequences? So they can illustrate this dilemma, how we go after something that might be human-specific and how we can do that in the future. Obviously, what one would like to do is take the chimpanzee version of FOXP2 then and put in transgenic humans. One would like the human FOXP2 and transgenic chimp and test their linguistic abilities. But there are ethics committees and I don't know what that you have to deal with. So it's not just a joke, though. This is, of course, what we would do if we studied Drosophila or mice to prove our point. And how do we get away from just speculating in this field? Because I think something we can do in the future is to hope to identify back mutations, so to say, in humans today. We know that the human genome is a small enough place and there are enough humans that every mutation compatible with human life is out there in the population, but in very low frequencies. But if we can genotype or sequence really millions and millions of people for hardly any cost, I think it will become ethically possible in the future to identify individuals with this type of back mutations. In essence, that's already done. You screen these FOXP2 mutations in families of speech mutations today. Right? You can also study this in cell culture, human and chimp versions. But what I sort of want to make propaganda for is to, after all, come to some animal model. And then it's, of course, a mouse that offers itself. And it's particularly easy here because, as we said, it's a very conserved protein. So humans differ from mice in only 3 amino acids. One is a very conserved amino acid here that we hope has no function. The 2 other ones is the ones that differ from the chimp. So to humanize a mouse gene, you just need to exchange this exon. You do that, of course, in embryonic stem cells by homologous recombination, and you end up having then a mouse that's from its endogenous FOXP2 gene now makes the human protein. So then you have the next dilemma, how you study a human-specific trait in the mouse. You try to speak to the mouse, and it doesn't help. So what we then ended up saying, this is a very conserved protein, so it could have something to do with something totally different than speech, of course, since it's conserved among all vertebrates, actually. So we worked together with something called the Mouse Clinic outside Munich in Germany that has a health screen in transgenic mice. Much, much better than what you ever get when you go to your doctor. So just to give you a feeling for this, there is sort of X-ray bone density, metabolism, hearing, strength, startle response, hematology, immunology, and so on, blood pressure, eye function. Always comparing then the transgenic humanized mice to litter mates that are wild-type born by the same mothers, so they really have the same environment. All in all, they looked at 323 phenotypic traits in these mice. Many of them correlated, of course, not all independent of each other, but still a very thorough screen. And there were only 2 of these traits where you had a consistent difference between the humanized mice and their wild-type litter mates. The first one, I really have no idea what it has to mean. The humanized mice are slightly more cautious in a new environment. So you measure this by having the mice in a social group. You open the door, they can explore an open area. Mice feel safer along the walls. So the mice will tend to stay along the walls, but sometimes venture into the middle, whereas the humanized mice all the time stay along the walls. But this is not something they do the whole time. Already after 3-4 minutes, there's no difference anymore between the humanized mice and the wild-type mice. So it's not that they cannot go into the middle, they're just more cautious sort of in the early phase of this. I have no clue what this means at all. But the second phenotype more totally shocked me, and that is that they have altered vocalization, these mice. And you measure the vocalization at 2-week-old pups. You take them away from the nest. You have a microphone here, and they peep, and the mother come and bring them back into the nest. And we work with Julia Fischer in Göttingen, who's an expert on analyzing these sonograms. There are a lot of different features in these sonograms they can look at. If you compare the wild type to the humanized mice, there are many differences. Sometimes the heterozygotes look like it's co-dominant. Sometimes it looks like it's a more recessive thing. So there's, of course, no way to say if this vocalization is more human-like. But it, of course, supports this very specific change in vocalization, supports the idea that these 2 amino acid changes in FOXP2 somehow influence a muscle coordination or the oropharynx or something that might be important for articulation. So this is very stimulating, but the frustrating thing is, of course, that almost nothing is known about the neurobiology of mouse vocalization. But at least you have a model system now that you can begin to go after. You can, for example, take out neuronal precursor cells or stem cells in principle, differentiate them in vitro, and look at how the neurons grow out these extensions that will become axons and dendrites. And you can see that these humanized mice, when you compare to the wild littermates, grow out longer neurites. You also see that in the brain, that there are more long-range connections in the striatum, in the neurons. You can look at sort of electric activity of the neurons, something called synaptic plasticity, in this time long-term depression, where you give a high-frequency stimulation of the neurons, and you see how they become less excitable in the wild-type animals, and even more so in the humanized mice, which is a model for learning and memory. What we know now from this model is dendrites in the striatum, we have increased synaptic plasticity in the striatum, and what I haven't told you is we have decreased dopamine levels in the brain. So you can begin to make hypotheses now what this might be about. And one idea is then that it has to do with these cortico-ganglial circuits, where the basal ganglia receives input from the cortex and the brainstem and gives output back to the cortex. So you at least can now start go after these human-specific things in a model that you can actually study. So this is quite stimulating, I think, and promising for a number of genes that have to do with brain development and function, where you have evidence for positive selection on the human lineage, where you can now make this type of mouse models. So finally then, what else can we expect in the next near future in this field? And I think one thing is actually genomes from other hominid forms other than Neanderthals. So we talked a lot about modern humans, about Neanderthals, but there are of course a lot of other hominids that paleontologists find, and we really know nothing or almost nothing about their relationship to Neanderthals and present-day humans. So we were very lucky last year then when we got a sample from southern Siberia from Professor Derevyanko, found in this cave in the Altai Mountains, and was a tip of a little finger of a human. Our question was really, was this Neanderthal or modern humans? So we extracted DNA, made a sequencing library, and with a technology developed in the lab, pulled out all mitochondrial fragments and found a lot of such fragments. So genome with 156-fold coverage, very high accuracy. Genomes difference, the biggest amount of differences we have is a bit over 100 genes. Neanderthals to humans today around 200. And to our big surprise, the number of differences from humans today to this person from Siberia is almost 400. So if we estimate the time to common ancestor here for Neanderthal and present-day human mitochondrial DNA, it's about 500,000 years. For this Denisova individuals, about twice as deep, about 1 million years ago. So it's of course very interesting what this individual is. We call it X at the moment in the lab, since we really don't know. And this is just now the mitochondrial picture here. The nuclear genome is sort of what we are now studying and looking at the relationships here, what it is like, if it's indeed a deeper divergence or how it's related to this. So I think to end up then, I think to then understand recent human evolution, what makes fully modern humans possible, what we need are genomes of our closest relatives, which are Neandertals, and then other human forms that we will get even better in the next few years. But it's not enough with that. We really need functional models also to test these candidate substitutions that we identify. And what really offers itself, I think, are really humanized mice there. So I should say that this is a big project, involves very many groups. It started as a very young collaboration with 454 Life Sciences. I should mention particularly Professor team that excavates in Spain and where we get samples directions now. This have involved even more people. I just want to stress that here at the meeting, Janet Kelso and Martin Kircher are absolute bioinformatic analysis of these genomes. Another crucial person was Ed Green, who really coordinated much of the bioinformatics in Leipzig and is now at UCSC, and was also featured yesterday in the keynote talk by Stephen Brenner. It's a very crucial guy, this. And population genetic analysis, particularly by David Reich here at the Broad, and Monte Slatkin at UC Berkeley. And before ending, I should also say that we do collect a lot of data from a lot of hominids that we need to analyze in the next year or two. So if you are interested in human evolution, you should really consider working with Neanderthals. Thank you for your attention.
Thank you for a fascinating talk. And now we know that Neanderthal knew how to play football, but not basketball, no? That's really, fantastic. And we have a small present.
Oh, thank you.
And we have, thank you, a few minutes for questions, please.
She was first.
Over there.
Hi, thanks for a wonderful talk. I was wondering towards what's next, given the fact that, you know, the difference is so small between genomes, and in the light of work like Rosemary Grant's on showing that at least part of Darwin's finches to differences in gene expression, whether you were considering going up coding variants.
Yes. You look at accelerated regions and things like that. Really, in the future, depend on understanding these non-coding variants today very well, so that one would have a chance to then, from sequence, understand some of it in Neanderthals, where we can't do experiments. Maybe it can just, as a curiosity point, Briggs, who was a graduate student in the lab, has shown that patterns at least survive in the Neanderthal DNA. See that by this sort of deamination, which methylated thing sort of preserves the teeth. But this is a problem of the bone, right, which is not the interesting tissue, maybe.
A question about the biogeography. Did they derive from a previous humans, human ancestors into Africa, or did they appear in Africa and then move out?
They must come from Africa at some point. Common ancestor humans is presumably in Africa, somewhere 3-4 years ago or so. But the morphology that paleoidentified Neanderthals is never seen in Africa. It's really some in Europe and the Middle East and Western Asia. The furthest east is in Uzbekistan, southern idea. And the earlier the Neanderthal morphology also becomes more extreme. Neanderthals are the most extreme in their morphology. The earliest Neanderthals, you call them Neanderthals, are in fact even a bit more like us.
So, selection, one always is careful not to confuse it with biome. Was that difficult for this project selection versus biostring conversion?
There is, yeah, so we looked at that. This substitutions we see is not this famous pattern of going from A to E, for example.
I see.
And it's really this derived alleles that we look at positions where we vary today. I don't think that these regions are subject to that. One can improve this selection screen by much better how you accumulate differences here, that they will become more now done.
Thank you.
Hi, I'm wondering whether you looked at any evidence of structural variation or copy number variation in Neanderthal genome.
Yes. So yes, Evan Eichler's group in Seattle have that, and it's reported in the paper. There are really no insights. There are slightly more than most humans are from each other, but very human-like. This low coverage, you really only can see big segmental duplicates or so you can't look at small things.
If there are no more questions, I would like to thank again the speaker.